This experiment, conducted by Messelson and Stahl, elegantly demonstrated that DNA replication is semi-conservative. The E. coli bacteria were first grown in heavy nitrogen, N15, making their DNA denser. Switching to normal nitrogen, N14, and allowing the bacteria to replicate once, resulted in DNA with an intermediate density, seen here in the centrifugation results. This proved that each new DNA molecule inherits one original parental strand and one newly synthesized strand. As we see in the diagram, the double helix unwinds, and each strand acts as a template for building a new complementary strand, perfectly illustrating the semi-conservative mechanism. Bacterial DNA replication begins at a specific site called the origin of replication, or ORI. The DNA double helix unwinds here, forming a replication bubble. Notice the two replication forks moving in opposite directions. This is what we call bidirectional replication. At each fork, DNA polymerase synthesizes new DNA. The leading strand is synthesized continuously, while the lagging strand is constructed in short segments, called Okazaki fragments. Replication proceeds until the two forks meet at the terminus region. Eukaryotic DNA replication, while similar to bacterial replication, involves a more complex initiation process. The origin recognition complex, or ORC, binds to the origin and recruits other proteins like CDC6 and CDT1. These, in turn, load the MCM complex, which acts as the helicase, unwinding the DNA. Now, look at the key players. DNA polymerase synthesizes new DNA, primase creates RNA primers, topoisomerase relieves tension ahead of the fork, and ligase joins Okazaki fragments. This coordinated effort ensures accurate duplication of the genome. Let's zoom in on the action at the replication fork. See how the leading strand is synthesized continuously in the 5' to 3' direction? The lagging strand, however, is synthesized in short Okazaki fragments, also in the 5' to 3' direction but moving away from the fork. Each fragment requires a primer laid down by primase. This difference arises because DNA polymerase can only synthesize in one direction, and the two DNA strands run antiparallel to each other. This slide highlights the essential role of the sliding clamp, pCNA, in eukaryotic replication. pCNA encircles the DNA, tethering DNA polymerase delta to the lagging strand and polymerase epsilon to the leading strand. This enhances the processivity of the polymerases, allowing them to synthesize long stretches of DNA without detaching. We also see other essential enzymes like primase, helicase, and DNA polymerase alpha, which initiates DNA synthesis. Finally, RNase H removes the RNA primers, and ligase seals the gaps between Okazaki fragments, creating a continuous DNA strand.